Hi everyone. Thanks to LabRoots for inviting me as a speaker to talk during this Genetic Week event. My name is Sairam Behera. I'm a postdoc research fellow at Sedlazeck Lab at Human Genome Sequencing Center of Baylor College of Medicine. So today I'm going to talk about our recent work on improving genomic analysis by fixing the human reference genome. So with the advent of sequencing technologies, the NGS data analysis has been central to most of the genomic research, be it plants research or the human genomic research. So we get all the high throughput sequencing data. Then during the analysis, we have to map them to a reference genome. And after that, we do further downstream analysis, for example, identifying the genomic variants, or it could be the gene expression analysis if the RNA-seq data sets, and many more analysis we usually do. So choosing a good reference or using a good reference genome is central to most of this analysis and as well as the genomic research as whole. So I will be mostly talking about the human reference genome. So these are some of the basic facts about the human genome. I think all of us must be knowing. So our human genome is encoded as DNA within these 23 chromosome pairs, and it has almost 3 billion base pairs. And it contains all the information about human development, physiology, disease, evolution, and many more stuffs. And we all know that in late 1990s, we had this great project called Human Genome Project that started to sequence the whole genome. And that included 6 different countries and 20 different labs, including our Human Genome Sequencing Center here at Baylor College of Medicine. And by 2001, we had this paper in Nature that talks about the initial draft of the human genome reference. And by 2003, it was announced that the human genome reference, human genome sequencing project has been completed. And by that time, it was 99% of the genome's gene-containing regions were finished with a very high accuracy rate. But if we talk about the whole genome, this telomere to telomere, so at that time it was 92% was completed. The remaining 8% was really the complex regions. They were highly repetitive, and technology was one of the constraints during that time. But some of those regions also included the coding, protein coding genes. So the current version that we all of using right now is the GRCS38, commonly known as SG38. And many of us are still using also the previous version, which is GRCS37 or SG19. This current version, GRCS38, has almost 3.2 billion base pairs, and that includes 20,000 protein coding genes. And last year, we all know that we had this great news about the completion of the Human Genome Project. And thanks to NIH and T2T consortium's initiative, that we had a new reference genome that we call T2T CHM13 or simply CHM13. That's the current version, is version 2.0 right now. It basically included almost 200 million new base pair as we compare to GRCS38 reference genome. And it has almost 2,000 candidate genes that really needs further study because that includes almost 100 genes. Potentially, they are the protein coding genes. And in 2021, a GRC consortium that basically maintains these GRC references, that put out a blog post, and they mentioned about some of the errors in the current GRCS38 reference genome. So one of the error is the false duplication in chromosome 21. That included some of the very important genes like CBS, U2, AF1. And also there are some sequences that has been contaminated with non-human sequences, but that was negligible. That's only 0.02%. So the two papers and below, basically, they talks about the false duplication error as well as the sequences that has been contaminated. So now we have, two references, the CHM13 by T2T and the GRCS38. So the question is that which one we have to use in future for all our research. And GRC consortium has already put out a notification that they are not going to have any future release, any future coordinate release. That means the GRCS39 or any other release. So the next question is that if we can totally replace GRCS38 with this CHM13. The answer is no at this time because we have lot of resources that is available with GRCS38 are not available or not are in the initial stages for the T2T CHM13, like gene annotation and some other stuffs. And also we have lots of data that has been already analyzed using the GRCS38. If you talk about any cohort or any population level analysis, we have humongous amount of data that has already been analyzed with GRCS38. And it will take a lot of effort, in terms of scalability, to realign all of them to CHM13 to do further analysis. So the question is that, is there a solution to fix the errors in the GRCS38, then do the realignment to GRCS38, but in a scalable manner? So with that, the recent work focused on these four things. The first thing, we have to identify what are the problems in the GRCS38, and then how to fix those problems, and then subsequently we have to see whether it basically affects the coverage, mapping quality, and subsequently the variant calls. And one of the statement we can say that as the reference genome has some errors, that means all the analysis that has been done previously, and if those analysis included those erroneous regions, that means they are not fully accurate. So with that, we first try to identify what are the problems in the GRCS38 reference. The first problem is the falsely duplicated regions. So here is a toy example that is showing the comparison between T2T reference and GRCS38 reference. So when I say falsely duplicated, that means there is a copy that has been duplicated in the GRCS38 reference that should not be there. The second error is the falsely collapsed regions. That's kind of opposite of falsely duplicated errors, where we have some of the regions that have been collapsed into one region in GRCS38. Again, this toy example is showing that T2T reference has two regions, but they have been collapsed into one in the GRCS38 reference. So all these errors, they are not the rare alleles, because we can say that two references are coming from maybe two different individuals, so that might be a rare event. But we compare to multiple samples to confirm that this is not a rare allele. This is basically an error. So before going to my next few slides, let me just give the definition of the some of the commonly used terms in the mapping. So the first thing is the coverage. When we say coverage, it's basically the number of sequencing reads that uniquely map to the reference or maybe cover a known part of the genome. And the mapping quality is basically the confidence score that read is correctly mapped to some of the coordinates. And when we talk about zero mapping quality, that basically indicates the sequences that are mapped to multiple locations in the genome. And finally, the average mapping quality is basically the mapping scores divided by all the number of bases in that region. So for the errors in the GRCS38, we looked into this NOMAD database, where it has almost 76,000 genomes from diverse populations. We looked into this particular gene called CBS that has been impacted by these falsely duplicated errors. Then we saw that there is a very low coverage of that, because as I explained before, it has a false copy. That means sequences that are mapped to the two different regions. It is mapped to the original as well as it is mapped to the false copy as well. So that might be causing this low coverage in that region. Similarly, for this gene called gprint2 that has been impacted by these falsely collapsed errors, there we found unexpectedly high coverage in that region. That's again because of some regions are collapsed into one, so more and more number of reads are mapping to that region. They should be mapped to different regions. So we also looked into some of the gene regions using this SG002 samples. So in this particular IGB screenshot that I'm showing, you can see in the panel 1, there are a lot of white spaces that indicates the zero mapping quality. When we align the sequences to the GRCS38 reference genome. And that also translated into almost no variant calls. You can see in the track that shows the SNPs. But if we fix the errors, that we are expecting a good mapping that I am showing you in the panel 2, and also some of the good variant calls that again I am showing in the track that is on the SNP, that is indicated by the SNPs. Similarly, if we talk about the collapsed regions, I'm showing you a gene called MAP2K3, where I extracted the sequences from the HG002 sample, then I mapped to the original reference genome. Then you can see in the panel 1, where it says the HG002 original mapping, you can see a very noisy mapping there. And that translates into a noisy variant calls that you can see on the track that shows the variants. But if we fix those errors, we should be expecting a less noisy as well as less noisy variant calls and less noisy the mapping. And whenever I say less noisy, that means it should be the true variant calls, that we should be eliminating all the artificial or the incorrect variants. So with that, we know we have some issues with the GRCS38, and we have almost 1.2 megabase pair of falsely duplicated regions and 8.04 megabase pair of falsely collapsed regions. So the bottom right figure basically shows the regions in the chromosome where this erroneous events has been occurred. The greens basically indicates the duplicated and blue indicates the collapsed regions. And here is another toy example that I'm showing. That's the comparison between GRCS38 and CHM38 in reference that shows different errors. And these errors basically impacted 33 protein coding genes, and that includes 12 with medically relevant genes. And some of the genes that are impacted by duplicated are GPRIN2, DOPS22, and for the genes that are impacted by collapsed errors are MAP2K3, KCNG18, and some other genes as well. So the next question is how to fix these problems. So for the falsely duplicated regions, one simple fix is to mask those regions that has been falsely duplicated. So that means you can put NNN in those regions that are the erroneous regions. But for the collapsed regions, as two different regions or more than two different regions have been collapsed into one, we could put a contig sequence or the extra contig in our reference. In this example, you can see there are two regions has been collapsed. We can put the region that has been collapsed into the reference as a decoy contigs. So by doing that, we are not disturbing the coordinates. So any further analysis on this reference will not affect the coordinate related analysis. So all the decoys that we have added during the fixing of this GRCS38 reference are coming from T to T CSM13 reference. These are basically the regions where we found they have a low KMR based copy number as compared to CSM13. And we also validated this with almost 99% of the samples in the Simon's Genome Diversity Project. This was done because to make sure these are not the rare alleles, these are really the errors in the GRCS38 reference. And these decoys, they represent either part of the gene or the full gene, or it could be a pseudo gene. And also there are some gene fragments, they don't have any annotations. And with this, basically, we fixed the erroneous GRCS38 reference. We generated a new modified reference that contains 24 duplicated regions that has been matched, and also 27 collapse regions for which we added the decoys. And recently, in that means in 2022, GRC Consortium has released a blog post where they have given a bed regions that can be used for any analysis, and those bed regions basically corresponds to the duplicated errors that we are working on. And with fixing this reference, then our next job is to show whether it is really correct or not. So one thing we have to assume that if we resolve these regions correctly, then it should impact the mapping quality. So we did some analysis for that, but before that, we basically developed a tool to do the remapping. This is because if we do the remapping of all the sequences again to the whole genome, the whole reference genome, in our case the modified reference genome, then it will not be scalable. That means the cost will be same as remapping to the CSM13 reference, and the scalability for the large consortium or the population analysis will not be that good. Because the alignment step in this process is always a time consuming step. So because of that, we developed a tool called FixItFelix. For that, what we did, we only selected the BAM regions that are impacted due to these errors, as well as to avoid the edge cases, we also extracted the BAM regions in the downstream and upstream regions by taking 5 kilobase pairs of them. And then we used the SAM tools as well as the mapping methods to align them to the modified reference genome. And here we have different modules for the short read, long read, as well as the RNA-seq datasets, because we use different mapping methods for different sequencing technologies. And surprisingly, this takes a very less time. It's less than 5 minutes to do an analysis for a 40x coverage BAM file. Again, this is because we are only focusing on the small regions that are impacted due to the errors. But then basically we wanted to make sure that this realignment process, realignment strategy is as good as by doing the remapping to the whole genome. So we have to make sure that we are not losing any information by doing this. So the first experiment we did using this SG002 sample, which has been widely studied, we wanted to see how the mapping quality is affected when we use our modified reference genome. So what we did actually, we compared the average mapping quality for all the regions that are impacted due to these falsely duplicated errors. Then we used the original BAM file, that means the sequences that are mapped to the original reference, as well as our BAM file where we use our FixItFelix tool and the modified reference genome. The green bars basically showing the FixItFelix with the modified reference, and you can see almost all regions we saw a significant improvement. And if we take all the regions together, we saw that genome mapping quality has been decreased by almost 72%. And the x-axis, basically, I am showing the gene names that are present in those erroneous regions. Some of the genes are denoted by a star. That means there are multiple genes present in those regions. Similarly, we did the same experiment for the collapsed regions, and we saw there are many regions where we saw a decreased mapping quality, and the zero mapping quality has been increased by 20%. But in a way, that is also good for us, because we wanted to make sure that by doing that, we are clearing the erroneous variants. So after doing some experiment on the mapping quality, then we wanted to focus on the variants. When I say variants, the small variants, including the SNPs, indels, as well as the structural variants. So first thing what we did, actually, we manually checked some of the genes that are impacted by these erroneous events. This particular gene called CRYAA, which is a medically relevant gene, and it's associated with many diseases like eye, oral, as well as neural diseases, and that's present in the chromosome 21. So here you can see in the first panel that shows the SG002 original mapping. That means sequences from the SG002 sample has been mapped to the original reference genome. You can see there are a lot of white spaces indicating the zero mapping quality. Then you can see the red triangle that I'm showing for the SNPs. There is almost no variants in those regions. But when we used our modified reference genome, you can see a good mapping quality, as well as there are some variant calls. And for the mapping quality, we also tried some other samples. We took 3 other samples from this ARI cohort. Then we saw the mappings are consistent with SG002 when we used our modified reference genome. Similarly, we also looked into another gene, CBS, which is also medically relevant gene. And we saw the similar effect when we used our modified reference genome. You can see the mapping quality has been improved using the remapping strategy. When I say remapping, that means using our FixItFelix tool as the modified reference genome. Then you can see the green triangle basically shows more number of variant calls as compared to the red triangle on the SNP track in this IGV screenshot. And the mapping quality has been consistent for other samples as well. Similarly, we took another gene called MAP2K3, and this particular gene has been impacted due to falsely collapsed errors. Then you can see the panel which says SG002 original mapping. We have a very noisy mapping there, and that translates into noisy variant calls. But when we used our remapping strategy with this SG002 sample, then we saw a less noisy mapping as well as less number of variant calls. And again, the mapping quality has been consistent with other samples as well. So the next thing we wanted to make sure whatever the variants we have been calling or we are detecting, they should be the true variants. By improving the mapping quality, we should be calling the true variants. To test that, we used this benchmarking data sets that has been developed by this NIST and Genome in a Bottle team. We particularly focused on the challenging medically relevant gene truth set. That's called the CMRG version 1.0 truth set. The first analysis we did the variant calling for the original mapping. When I say original mapping, that means the original sequences, the sequences mapped to the original GRCS 38 reference. Then also we did mapped to the modified reference using our FixItFelix tool, as well as we extracted all the sequences and we did a global remapping to the modified reference genome. In this particular plot, I am showing the F measures of the SNPs and indel calls with the original reference genome, with the modified reference genome. And for the modified reference genome, we used both the strategies using our FixItFelix tool as well as the global remapping. You can see the green and purple bars showing the results. They are showing basically almost F measure score of 1. That means perfect recall and perfect precision for the SNPs as well as indels. And they are consistent for both FixItFelix tool as well as global remapping. But if you compare it with the original mapping, that means with the original reference genome, you can see a significant improvement for the SNP calls, and for the indels, there are almost no calls for the original reference, but we have almost precision score of 1 for indels. Similarly, we used the collapse regions as well to see how this modified reference is improving the variant calls. We didn't see that significant variant calls as we saw in the falsely collapsed regions, but we see some improvements in the variant calls, both for SNPs and indels. Again, for the modified reference, we are using the FixItFelix tool, do the remapping strategy, and the global remapping. That means we are extracting all the sequences and mapping to the whole modified reference genome. With that, actually, we saw there is some improvement in the whole genome sequencing data sets. Then we wanted to focus on the whole exome sequencing data set because we wanted to see the variants in the protein coding regions as well. And we took this SG002 samples whole exome sequencing data set, and we did the similar kind of analysis, the variant calls, and compared with the truth set. Then we found there is a significant improvement when we used our FixItFelix tool combined with the modified reference. Again, you can see all these green bars that's indicating the significant improvements as compared to those red bars that was due to the original reference genome. And we tried for both duplicated and collapsed regions, and we verified the SNPs as well as indels, and we saw there is an improvement for the variant callings for this whole exome sequencing data set as well. So as we saw the improvement in the variant calling is the SG002 sample, then our next question is that whether it is specific to SG002 sample or not, because we all know this SG002 sample is coming from a European individual. So we wanted to make sure that it is not specific to a particular ethnicities. So for that, we wanted to do some experiment on the samples that's coming from various ethnicities. But one challenge we had that we didn't have any truth set, just like SG002. So because of that, what we did, we used this DeepCall tool to generate a benchmark dataset. That benchmark dataset is not as good as the one developed by the Genome in a Bottle team, but that is enough for doing some validation for our case. And DeepCall basically uses the haplotype result assemblies. Those assemblies are basically coming from the long read assemblies. It is the hi-fi based assemblies that we have used for this particular analysis. And using the DeepCall tool, we created the benchmark dataset. Basically, DeepCall calls all the variants using this hi-fi based assemblies, and we used that benchmark set to do further analysis. But before that, we wanted to make sure that this DeepCall tool works as good as CMRG tool for SG002 dataset. Because the good thing is that CMRG benchmark also use DeepCall results for doing the benchmarking. Because of that, we just compared the CMRG variant call sets with these difficult variant call sets, and we focused only on those medically challenged regions. And we found that there is a perfect concordance for the variant calls, except there are some aberrations in the indel calls. But above all, actually, we trusted that this could be as good as the CMRG benchmark set. With that, we did some analysis for the SG002 sample first. Then we again, verified the variant calls in each and every regions that are impacted by these duplicated errors. Then we see a significant improvement in all the cases. Similarly, we also did the similar analysis for the collapse regions as well. And you can see there are some regions where the variant call performance has been decreased. That's again because some of the genes are pretty complex, and that really need further studies. And with that, basically, we trusted the difficult benchmark set. So then we focused on our analysis on those different samples that's coming from various ethnicities. We selected these 8 samples from the T2T diversity panel, and we selected because for all these samples we had the hi-fi assembly available, so that we could run the DeepCal tool, and we can generate a variant call set, and that we can use as a benchmark set. And we did the similar analysis that we did for SG002. This plot basically showing all the variant calls combined together. The y-axis is showing the F measure. You can see all the green boxes are due to the modified reference genome that we used. And again, we used our FixItFelix tool to do the realignment to the modified reference genome. And all the red boxes are showing the variant call performance for the original reference. You can see for all the cases, the green boxes are always higher than the red boxes. That's showing the variant call improvements using the modified reference genome. And we also saw the similar patterns that we saw for the collapse regions as well. Whatever we see in the SG002 sample, we also saw the similar pattern for all the samples taken together for different ethnicities. So with that, we are confident that FixItFelix works for SG002 sample as well as the other sample. They are taken from different ethnicities, and the run time is very, very fast. It's in few minutes as compared to greater than 24 hours for the whole remapping. And that basically enables us to work on the large cohort. That means, the population data sets. So for that, basically, what we did, we tested our FixItFelix tool as well as our modified reference on almost 4,000 genomes. And those genomes basically 30x coverage, and they are taken from that 1,000 genome project dataset and some of the genomes taken from this human genome diversity project as well as the TopMed consortium. The first thing we wanted to see the coverage analysis, and you can see on this left figure that's showing the mean coverage for the whole genome. But when we concentrated on the regions that are impacted by the falsely duplicated errors, then you can see the low coverage when we take all the genomes together. But when we used our modified reference genome, that errors has been corrected. You can see the mean coverage is same as the whole genome that is showing on the top. Similarly, we also checked the mean coverage for the collapsed regions. Then you can see this is a high coverage for the collapsed regions when we took all the genomes together. But when we used our modified reference genome, that the coverage bias has been corrected. Similarly, we did the variant calls for all the data sets that we analyzed in this particular analysis, and we saw for the falsely duplicated regions, we saw elevated rates of variants with heterozygous deficiency. That is indicated by the positive inbreeding coefficients here. Similarly, we saw excessive heterozygosity for the falsely collapsed regions. That's indicated by the negative inbreeding coefficients in this particular plot on the right side. But when we used our modified reference genome, those biases has been corrected. That you can see, that's almost consistent with the whole genome in this particular figure. So now we have the improvement on the single sample. We tested for the whole genome sequencing as well as the whole exome sequencing. We also tested for the population level by using the TopMed and 1000 Genome Project datasets. The next question is what about the RNA-seq datasets? Are there any erroneous eQTL signals, or can we correct them using this modifier reference? So we did some analysis using almost 450 RNA-seq datasets from the 1000 Genome Project. In this particular plot, you can see the count per million for all genes that are impacted due to these errors. The x-axis shows the count per million with respect to the original reference, and y-axis shows the count per million with respect to the modified reference. You can see the blue or the green bubbles on the top. That indicates the count per million has been increased because the false duplication errors has been fixed for the modified reference. And you can see some of the blue bubbles on the top of the x-axis. That's because some of the false duplicated regions which are present in the GRCS 38, that is not there in the modified reference. So there is no counts for the modified reference. And for the collapsed regions, we found only this DOPS22 gene has some substantial changes in the gene counts. And we did lots of analysis from the eQTL signal, and we found some of the eQTL signals has been corrected using the modified reference, and all this analysis is there in our publications. So with that, this is just a summary of our work, and we found the errors in the GRCS 38 reference that impacted almost 33 protein coding genes, including 12 are medically relevant genes. And all studies that has been done in this reference genome, if they included those erroneous regions, that means they reported some wrong eQTL studies as well as the analysis were wrong. So we corrected those errors and we generated a modified reference genome. And we saw that greatly improves the variant culling across different samples as well as different ethnicities. And we also provided a better remapping tool that can be used for short reads, long reads, or the RNA-seq data, and also it can scale the cohort analysis or any population analysis. Our tool is available in our GitHub page, and our publication that recently published in Genome Biology is there. I have given a link here. With that, I would like to thank all my colleagues who worked with me, including my postdoc mentor, Fiz, at Baylor College of Medicine, and my colleagues, Medhat, Luis, and Jesse, and Justin from Genome Portal team, and our collaborator from UC Davis and UC Michigan. Okay, thank you.